On behalf of the organizing committee, I'd like to welcome everyone joining today's section, cutting-edge technologies and their application in neuronal biology. The advancement of technology apparently played a central role in the research biology, including neuronal biology. Cutting-edge technology have profound impact on both basic research and the therapeutic applications. These include single-cell sequencing, gene editing, spatial transcriptome technology, and multi-omics technology, and so on and so forth. Basically, the emergence of new technologies have really made it possible for researchers to explore new frontiers and have really promoted serious advances in the field of biology, of course, including neuroscience. So in today's section, we have 4 renowned scientists and 1 4 speakers who will share with us their latest progress in research. So without further ado, I will hand this over to Professor Tang to host today's section. So Fuchou, your turn.
Okay, thanks. Thanks, Wensheng. I think this is really a great honor. So for this section, it's organized by Professor Wensheng Wei, Professor Xiaoqun Wang, and me. I'm from Peking University. I'm Fuchou Tang. Let me first introduce our first speaker, Professor Bing Ren. Dr. Ren is the director of the Center of Epigenomics, and he's also a professor of cellular and molecular medicine at the University of California, San Diego. Bing is also a member of the Ludwig Institute for Cancer Research. And Bing is studying how the non-coding sequences in the human genome direct the spatial and temporal patterns of gene expression, and how changes in these sequences contribute to human diseases. I think I want to take this chance to say something more about the Bing's work. I think Bing's work is really a pioneer work, and it's a really combination of beautiful new technologies and new biological ideas, and really give us a lot of new insight of the developmental biology and other biological field. And I think personally, traditional biologists is very like the way to do the research about gene expression. For example, for one important gene or transcription factor, you're trying to find 1 or 2 key targets of the gene. And then for this target gene, you further try to find 1 or 2 key downstream gene. But I think that's not the real regulation way in our human cells. So in fact, in our human cells, for example, for one transcription factor, usually it parallel regulate maybe 500 or even 2,000 downstream targets. And these every target usually also regulate downstream another dozens or thousands of targets. So this is the beauty of our cell. And you can always try to use the traditional way to say, well, let's imagine we just one gene and 1 or 2 key targets. But in the real cell, it's always one gene, one transcription factor regulate hundreds or even thousands of targets. But how these thousands of targets in parallel is regulated by one transcription factor, and then it's still not in chaos, everything in order, and makes the cell progress so well. So I think Bing contribute a lot to this point of the regulation of gene expression in our cell. So I think in the future, I think with the beautiful work from Bing's lab and also other labs, so very likely we will have a much better understanding, or maybe we change our mind of the gene regulation and how it connects to the phenotype of our cells and of our individuals. So I think let's welcome Bing.
Thank you so much, Fuchou, for that very kind introduction. I'm glad to use this opportunity to share with everyone research going on in our lab. The title of my talk is about single-cell analysis of epigenome in human and mouse brains. I have a couple of disclosure to make. I'm a co-founder of Epigenome Technologies and RIMA Genomics. The main motivation for me to carry out single cell epigenomic analysis in brains is based upon this hard fact. Neuropsychiatric disorders are one of the leading causes of disease burden in the world. Millions of people are suffering from mental illness, neurodegenerative disorders such as Alzheimer's disease every day. And there's no effective cure available to treat these disorders. And that is one of the urgent needs that we face in the modern society. And to develop cures and treatment, what we need is a better understanding of the disease mechanisms of such illness. Genome sequencing technologies have revealed many DNA variants as likely risk variants for Alzheimer's disease or schizophrenia. But 90% of such variants are non-protein coding, and their function are hard to interpret. And that is one of the major hurdles that we face. A second hurdle is that many of the neurodegenerative disease is age associated. Aging is a major risk factor for such illness. How does age contribute to such illness is unknown, and is one of the major questions that we also face. So today I'd like to demonstrate how we use single cell epigenome analysis to advance in both these fronts. The major paradigm that we now operate is that much of the human disease is contributed by not protein coding sequences, but rather non-coding DNA sequence. These variants act by blocking or disrupting transcription factor binding to DNA, and therefore affect transcription of the target genes in disease-relevant cell types. It is estimated that more than 50% of the non-coding risk variants may act this fashion. But we still don't know where about the transcription regulatory elements are, when and where they act. We also lack the information about the target genes controlled by such element. And further, how risk variants affect transcription factor binding and which transcription factors are affected is largely unknown. So these are the major barriers that we face. Today, I'd like to convince you or propose that by using epigenome analysis, we can begin to understand genome regulation in different cell types and to begin to decipher the non-coding variants in the genome. First, I'd like to introduce the concept of epigenome. This refers to the modifications to DNA, such as DNA methylation, or histone proteins, such as methylation of lysines or acylation of lysines, and so on, that can be propagated in each cell and influence transcription of the genes in different cell types in a sequence invariant fashion. In other words, transcription of the genes is impacted not only by the DNA sequence, but also by cell type specific modifications to the DNA and to histone proteins. And further, the chromatin structure in three-dimensional space also are playing an important role in the gene regulation. So altogether, these molecular modalities contribute to our to the transcription programs within each cell. And we can study them thanks to the development of high-throughput genomic technologies, initially by using chip-on-chip, we can discern the locations of histone modifications and transcription factors. More recently, DNA sequencing-based applications such as ChIP-seq, ATAC-seq, whole genome bisulfite sequencing, and Hi-C also allowed us to interrogate a genome-wide epigenome profiles in different cell types. So these early studies, many from our labs, has contributed to the understanding of enhanced location identification of cis-regulatory elements such as promoters, enhancers, insulator sequences in different cell types and tissues, and have been utilized by consortia such as ENCODE, Epigenome consortium to annotate the functional element in our genome. This previous method, though, worked on bulk tissues and are limited by cell types and tissues where you can obtain relatively pure population. And the heterogeneity of complex tissues present a major roadblocks for application of such methods. In recent years, single-cell epigenome technologies have made a great stride in this field. Now we have techniques to identify genome-wide methylation profiles, chromatin accessibility, histone modifications, and 3D genome confirmation in individual cells in a massively parallel fashion. This single-cell analysis can then be used to can be analyzed first by digitally grouping them based on how similar they are, and then these so-called clusters can be used to infer the cell type specific epigenome landscapes within each cell type. There are three major technology platforms that have been used to interrogate epigenome in individual cells. One of the first utilize miniaturized assays in test tubes or 96-well plates or micro-well plates. And these are essentially modest or low throughput and high cost. Droplet-based assays provide a convenient and cost-effective fashion to analyze epigenome in individual cells. 10x genomics or Bio-Rad, for example, all use droplet-based strategy where you can encapsulate individual cells into a single droplets and add barcodes to individual cells, DNA or RNA molecules that will result in single cell-based profiles. And more recently, a split and pooling, or also known as combinatorial indexing strategy, has been introduced. In this strategy, you can basically perform multi-well assays. Each well, instead of the individual cell, you can put thousands of cells in individual wells. This then combined together, split into a different multi-well plates where you add a second round of barcodes. So you can do this multiple runs, creating exponential number of DNA barcodes for you to keep track of individual cells. So this is extremely high throughput and can be customized to different applications. So my lab 5 years ago have adopted the combinatorial barcoding strategy. We first apply this strategy to interrogate open chromatin in cells. So we, through automation, have been able to optimize such single cell ATAC-seq strategy to interrogate open chromatins in frozen tissue samples. We achieved high throughput, 10,000 cells per sample, relatively high coverage, and very low cost. And we have achieved a very high success rate as well, achieving 98% success rate. So this strategy is now used in our production effort. Additionally, we also have implemented, actually invented a strategy for analyzing single-cell attack-seq data. This is known as a SNAP attack. The idea is that you take the data from individual cells. You first group them into clusters using dimension reduction strategies. And these clusters are then annotated based upon what type of cell markers they carry out. And then we perform peak calling and identify differential peaks and infer master transcription regulators and even infer likely candidate target genes for each regulatory elements. So this complete package of a single cell attack-seq analysis was made available several years ago, and now we continue to improve upon it. The second version of this was recently released on GitHub, and you can find the source by just using this 2D barcodes. So using this pipeline of single cell attack-seq, both experimental and computational pipeline, we have analyzed first mouse brain and then human brains, and as well as many additional tissue. Applying this to mouse cerebrum, we obtained single cell attack-seq profiles from about 800,000 cells. These include the olfactory lobe, isocortex, hippocampal formation, striatum, and pallidum. So this, by clustering, we were able to identify 43 major types and 160 cell types that come from the non-neuronal cells. These are the inhibitory neurons and then the excitatory neurons. We annotated each cluster based upon known marker genes. When you have a marker gene, you can see that a promoter of that gene typically is highly accessible in cells where it is active and not so much where it's not active. So based on this kind of a marker gene accessibility, we can annotate the cell type. And we found that our annotation based on the content accessibility in general matches that annotated using RNA-seq experiments. So giving us confidence that single cell attack-seq is able to deconvolute cell types as effectively as our single cell RNA-seq profiles. We now make this resource available to the general public through a website called Catalyst. It's short for CIS Regulatory Atlas. So we can basically explore the real raw data using this web resource. We can first identify regulatory elements based on the open chromatin landscape of each cell types. Altogether, we identify close to 500,000 potential regulatory elements that are operating in one or more of the 160 cell types in the mouse brain. And you can see from this heat map, the majority of such elements show lineage-specific accessibility, indicating that they are likely distal enhancers controlling transcription of the gene. So our atlas can be very useful for designing cell type specific enhancers. This is two examples from the Allen Institute where the accessibility in glutamatergic neurons or inhibitory neurons are used to define their cellular specificity And by using AAV-based enhancer reporter assays, these elements can be used to drive reporter gene in the expected cells. For example, this is a hippocampal neuron enhancer, and you can see in hippocampus, this is the region that are light up where the GIP is active, while the excitotomic neuron, layer 5 ET neuron specific enhancer is used to drive reporter in that specific cortical layer. So we believe that our atlas of chromatin accessibility can be useful for designing highly cell type specific enhancers, reporters. So after profiling the mouse brain, we also now have obtained a equivalent data sets for the human brain. This is in collaboration with Anne Lane from Allen Brain Institute, Joe Acker and Maga from Salk Institute. What we did is to interrogate 41 human brain regions from up to 3 donors each. This includes the major areas of the cortical layers, as well as cerebellum and midbrain and hindbrain. So from currently close to 600,000 cells profiled, we were able to group them into 120 cell types, including 22 glial cell types, 56 GABAergic, also known as excitotomic neurons, as well as 45 glutamatergic neurons, GABAergic inhibitor neurons, and glutamatergic excitotomic neurons. And again, we annotated the identity of each cell cluster based on the accessibility of the marker gene promoter, and then determine the likely regulatory element within each cell type. So again, like what we've shown in the mouse cerebellum, we were able to identify a total of 442,000 candidate regulatory elements that are active in one or more of the human brain cell types. And we show again that the majority of these elements are accessible only in selective neuronal or glial cell lineages. Altogether, these elements adds up to roughly 13.7% of the human genome. Majority of them are localized to either intergenic or intronic regions, but some of them are localized to repetitive DNA sequences such as line element and sign elements or LTR element. We next want to ask whether this atlas of cis-regulatory elements can help us determine disease-relevant cell types. We use a computational framework called linkage disequilibrium score regression analysis to do this. So this graph shows how genome-wide association studies help us identify the SNPs or the sequence variants that are genetically associated with certain traits or disease. But as you notice, the reported leading or tagging SNPs are not the only one that likely contribute to the genetic heritability. For every SNP, there are dozens of others that are equivalent because they are in so-called linkage disequilibrium blocks. They are inherited in the same fashion as the leading SNP. So just based on the genome-wide association study, you are not able to identify which one is causal, which one is not causal. However, we can use functional stratification based on the cell type specific regulatory elements to stratify this sequence variants. We assume that if the sequence variants are contributing to a disease phenotype by disrupting regulatory elements, then the cell type where it's relevant to the disease is likely going to have enhancer landscape highly enriched for SNPs that are either in the leading SNP or in LD block with the leading tagging SNP. So this framework essentially assumes that SNPs are in LD with a functional category and essentially determine enrichment of particular functional category in the GWAS SNPs. So the enrichment is then can be reported in this heat map, where the color represent highly enriched cell types and disease association. So this is roughly 30 different neurological disease or traits against 123 brain cell types. And you can basically see that for some traits, such as Alzheimer's disease, there is only 1 or 2 cell types strongly associated with it, and that is microglia cells, different types, subtypes of microglia cells. For schizophrenia, there is a large number of neuronal cell types and are strongly associated with this condition. Intelligence, similarly, you can see a large number of neuronal cell types, both excitory neurons as well as inhibitory neurons can be found in this fashion. So this provided an immediate advance in our understanding of many neurological disorders. Let me give you one example. As I mentioned, Alzheimer's disease is strongly associated with microglia enhancers. One of such enhancer near the BIN1 gene harbor many risk variants for Alzheimer's disease. In fact, BIN1 is a locus that are strongly, probably the second most risk locus for Alzheimer's disease, especially late onset Alzheimer's disease. And one of such risk variants happened to fall within the enhancer of microglia, and in collaboration with Chris Glass lab at UCSD, basically this enhancer is now shown to be responsible for BIN1's gene expression exclusively in microglia cells, but not in neurons or astrocytes. This basically suggests that regulation of BIN1, specifically in the microglia cells, is likely responsible for the phenotype for its role in Alzheimer's disease. Just to summarize what I've just illustrated, we were able to produce single-cell analysis of chromatin accessibility for both mouse and human brains. This enabled us to annotate candidate regulatory elements in the human genome that are involved in regulating brain-specific expression. We uncovered cell types related to neurological disorders, such as schizophrenia, Alzheimer's disease, autism, and many others. We demonstrate that the single-cell epigenomics is very useful in improving our understanding of polygenic human traits and clinical relevant therapeutics targets. So genetic factors is only one contributor to neurological disorder. Environmental factors and aging is another major risk factor for many neurodegenerative disease, especially Alzheimer's disease. The risk of developing this dreadful disease increase by age. At 80 years old or older, the chance of developing Alzheimer's disease grow to 10 to 20% for men or women. So it's very important for us to understand how aging affect the disease progress, and to do that, we need to first understand what happened during aging, especially aging of the brain. Aging is a long-standing scientific question dating millions, thousands of years, millennium, in humans' quest. And over the years, it has now recognized that there are many factors contributing to aging. For example, in this review in 2013, many hallmarks of aging was reported. This include DNA genomic instability, stem cell exhaustion, cellular senescence, mitochondria dysfunction. But one of the aging hallmark that I'm particularly interested in is epigenetic alteration. As I mentioned, epigenome refers to the modification of DNA or histone proteins, and such modification usually is dynamic, and they control the cellular expression of genes. To better understand how epigenome changes during aging of animals or human beings, we use mouse as a model system. This is carried out in collaboration with Xiangming Xu from UC Irvine. We focus on 5 major organs in the mouse, including heart, bone marrow, skeletal muscle, frontal cortex, and the dorsal hippocampus. And we perform single-cell ATAC-seq as well as single-cell RNA-seq to understand, to try to get a high-resolution picture of what changes in the chromatin during the aging process. From 3 age groups, 3 months, 10 months, and the 18 months old mice, we obtained single-cell ATAC-seq profiles from total 227,000 nuclei. Performing cell clustering, we grouped them into 32 major cell types and more than 60 subtypes in these tissues. We perform peak analysis from each of the chromatin, open chromatin landscape from each cell types. We were able to identify a total of 353 candidate regulatory elements, again, showing that most of them exhibit lineage-specific accessibility in the cells, in the mice. We are very interested in asking what changes occur in chromatin accessibility in each of the cell types during aging. We first grouped them into cell clusters. We found that most of the cell clusters don't change in abundance, but rather they exhibit age-dependent differences in accessibility. We identify such elements and determine their biological pathways. So altogether, we found 77,000 elements to show age-dependent accessibilities between 3 months and 18 months. So the top several cell type tissues that exhibit such changes are bone marrow and dorsal hippocampus, as well as frontal cortex. The genes that are changed, affected by such process can be determined. For example, in dorsal ganglia neurons, these are in the hippocampus, they show changes mainly in upregulated accessibility, and they're strongly enriched in transcription factor motifs, including FRA and JUN. These are AP1 stress factor, transcription factors that are typically involved in regulating neuronal activities. And we are finding, of course, the signaling pathways also affected by this. In skeletal muscle, what we discover is many elements show a reduced accessibility, and these are enriched for myoblast proliferation genes or pathways, suggesting that in older mice, skeletal muscles are losing their capability for regenerating myoblast. And this is regulated by signaling pathways or molecules such as STAT and STAT3 or STAT4 pathways. We also determined that changes in accessibility in by and large is associated with changes in gene expression. For example, ROBO1 is highly active in young animals, and they are reduced in expression in older animal, and this is associated with the loss of chromatin accessibility at this alternate promoter of ROBO1. What's interesting is that when we compared the elements that show age up or age down during the in different neuronal or non-neuronal cell types, the region heterochromatin show unexpectedly enrichment of those that gain accessibility in older mice. And this is especially true for excitatory neurons. As you can see, this box, this circle indicates strong enrichment for elements that are gaining accessibility in older mice. And this come from frontal cortex or dorsal hippocampus in a mouse. And this kind of enrichment is absent in inhibitory neurons or glia cells. And this is true not only for frontal cortex and hippocampus, but also in other brain regions, such as entorhinal cortex and part of the amygdala. This suggests that this type of heterochromatin enrichment of gained accessibility is a brain-wide phenomena. And looking further, we ask what they are. They tend to show, again, this is a couple of regions that are marked by heterochromatin marked lysine 9 trimethylation at the age of birth. And you can basically see that in 3 months, there's no accessibility in this region. 10 months, you can begin to see some accessible regions. In 18 months, this type of neuron have significantly gained accessibility. This is true for L2, L3 type of neurons in frontal cortex, also true for L4. But this is not seen in oligodendrocyte or VIP inhibitory neurons. This is another region where you can see gradual increase of accessibility over aging, and this is selectively found in excitatory neurons. The gain of accessibility in otherwise silent heterochromatin regions raise the possibility that we are seeing a loss of heterochromatin or erosion of heterochromatin in old excitatory neurons. To directly test this hypothesis, we perform an experiment known as pair tag. This is an assay where we can jointly analyze histone modification and RNA expression from the same individual cells as a high throughput manner. We published this last year. So using these techniques, we profiled both young and older frontal cortex and the dorsal hippocampus. We obtained neurons, many thousands of cells from these tissues. We grouped them into inhibitory neurons, excitatory neurons, as well as non-neuronal cell types. And when we focus on the heterochromatin in these regions, we indeed see a reduction of lysine 9 trimethylation exclusively in heterochromatin regions from the excitatory neuron, but not in glial cells, nor in inhibitory neurons. So in loss of lysine nitride methylation in these cells typically is accompanied by increased RNA transcript from these regions, suggesting that loss of heterochromatin actually resulted in activation of genes. This is true for many heterochromatin regions. This is a survey of all 400 some heterochromatin domains in all the cell types. What we are seeing is a large number of heterochromatin regions have lost lysine nitride methylation mark, and correspondingly, these are also gaining accessibility and RNA transcript levels. Of these regions, one class of repetitive DNA sequences jump out. This is called L1MA5A. This class of elements appear to significantly elevated their chromatin accessibility exclusively in the excitatory neurons, but we don't see that in inhibitory neurons or neuronal cells. What is line elements? Line elements are one of the most abundant repetitive DNA sequence element in the human genome or in the mouse genome. They are present in roughly 300,000 copies. They have they are remnant of the ancient retro elements. They consist of 2 ORFs and 5' UTR and 3' UTR. Those ORF proteins, without the mutation, would usually facilitate encode the envelope protein and the integrase for the particle to integrate and jump around. But 99% of the line element has now become inactivated due to mutation, so they don't jump around. But they can still make RNA and sometimes make proteins. So we indeed, using staining, immunostaining, we can detect elevated levels of ORF1 transcript, protein levels in older neurons compared to the young animals. So this suggests that line 1 is indeed reactivated. So when we have this study, there's a paper came out just actually a few days ago that suggests that line 1 RNA expression can cause heterochromatin erosion and actually can, if you inhibit such line 1 by antisense oligo, you actually can prolong lifespan in a progeroid mouse model, which is a premature aging model. So this is really interesting because now we are seeing a direct link between line 1 expression and the heterochromatin loss, and we're going to now investigate whether this is relevant in the context of mouse brain aging. To summarize this second part of the story, we now generated single-cell chromatin accessibility maps for brain, heart, skeletal muscle, bone marrow from the young and middle-aged and older mice. We saw a dramatic loss of heterochromatin during aging in specifically cortical and hippocampal excitatory neurons. The loss of heterochromatin is restricted to excitatory neurons in this time frame and are affecting multiple classes of repetitive DNA element. In particular, activation of line 1 is observed. And we suspect that activation of line 1 directly caused the loss of heterochromatin in the excitatory neuron and therefore present a potential therapeutic opportunity where suppression of this line 1 transcription might prevent heterochromatin loss and improve cognitive function in older mice. In conclusion, I'd like to acknowledge my colleagues and collaborators who have been instrumental in the both part of the work I presented. I particularly mentioned Yang Li's and who was computational biologist, formed a single cell analysis. Sebastian Prezel at the Center for Epigenomics was responsible for generating much of the single cell Alice maps. The second part of the work was carried out by Yanxiao Zhang, who is now at Westlake University, together with Louisa Amaral in collaboration again with Sebastian Prezel at the Center for Epigenomics at UCSD. Our collaborators I mentioned during the talk, and our funding agency who supported work. Happy to answer questions. Thank you for your attention.
Okay, thanks for the beautiful talk. So any questions?
Hi, Professor. Can you listen to me?
Yes.
Thank you for your report. Very interesting. And my name is Guo Yuqing. Came from Peking University, and I'm the student of Professor Tang Fuchou. I have a question. I know that you have done a lot of work on genome epigenetics. I want to know is the epigenetic state of a specific locus and is reversible. Can the epigenetic state of a locus be edited like gene editing? Thank you.
That's a great question. Indeed, epigenetics provide an attractive therapeutic model because they are reversible. DNA, unlike DNA changes, a specific locus of epigenetic in the genome. Reversal of epigenetic mark in specific locus is still challenging. It's doable, and through CRISPR techniques you can achieve that, but delivering of such changes to inside the body is still a very challenging. So that is one of the major bottlenecks we face. However, in pharmaceutical companies, strategies to do specific perturbation of epigenetic processes, blocking the enzymes activities, either writers or eraser or even rater, are now being explored for therapeutic purposes.
Thank you for your answer. I got it. I have another question. So how the data should be interpreted and integrated when the result of multiple omics data are inconsistent? We often meet the trouble. Thank you.
Sorry, I lost part of your sentence.
I think what he mean when you do the different single cell omics separately, so how you integrate the data.
Got it. Yeah, it's there are ways to do this integration. For example, we typically use some of the common tools such as Surat, where you can integrate single cell TAC-seq and single cell RNA together based upon the nearest neighbor scheme. There are also other related similar integration tools that have been we're using also. Liger is another that we have tried, and single cell fusion. So there's a variety of tools that achieve integration across modalities.
Thank you, Professor.
So any other questions? Okay, I have several questions. So your work is really beautiful. So during the aging, you see for some of the genes are downregulated in some specific cell type, right?
Yes.
So I wonder, you see, because you are single cell resolution, you see for these downregulated genes, it's downregulated in the same cell type, every individual cell, or just in some cells just disappear, but other cells are still there?
we don't see many cases where the cell population is decreased or disappeared. Most of the
I mean in the same cell type, you see some gene downregulated. So is it downregulated every cell, or is just
very good point. That's a good question. We tried very hard to look for such patterns. By and large, we are seeing we cannot at this moment distinguish between the two possibilities because of the lack of I would say statistical power. So I cannot answer that, but that's a very important question. When we have deeper sequencing data, we should be able to address it.
Okay, thanks. And another question is about the line 1. You see during the aging, some of them is reactivated, and so seems something behind it is just the RNA or the protein of this line 1 contribute to the, for example, loss of the hyperchromatin. But I wondered, do you see some cis-regular effect? For example, near some very important genes, some line 1 are specifically activated, and then it directly affects the nearby genes expression level.
Yeah, that's a very good possibility. Well, right now we're focusing on mainly heterochromatin, where you don't see many genes or protein coding genes. These are gene desert. And we try to look at transcripts that are turned on. Most transcripts are non-protein coding. They're not likely to be functional. So what we interpret is that it's the production of line 1 or repetitive DNA or RNA element that is likely going to present a so-called immune reactivity that will trigger interferon response pathways and cellular immunity.
And another question is about now the single cell omics or multi-omics, the sensitivity is not so ideal. So I'm wondering, for example, when you analyze the different aging mouse, for example, the 3 individuals or at 18 months, you see a lot of the difference between the individuals, or just the technology is still not powerful enough, but looks just for every 18 months mouse, the same cell type looks is nearly the same. You can't see the difference.
That's right. We unfortunately didn't analyze too many replicates. So we haven't been able to assess how much variability there is in between individuals. Okay.
So another question. Sorry, but this really is a great work. So for the work your work in the human samples. So how about the human brains samples quality? Affect a lot of the result, or is quite stable? So basically, for example, after death, after many hours you collected, it's still quite okay, or still it need to be really quite high quality sample?
Yeah, that's a very good point. Very good question too. We collaborate with at Lane's group, who where they have established a very I would say rigorous procedure where they were able to obtain brain post-mortem brain samples at a stage where the I would say the tissue exists in a state where our single-cell tag-seq and RNA-seq are performed at very, very high quality. This is essential because we have definitely seen variability of quality when, for example, the samples are collected or preserved not at the optimal state. So we typically have to check the ring number RNA quality, which is not perfect for ataxia, but does indicate a certain levels of preservation of the tissue that will inform us whether the high quality data will likely result from that.
Okay. Still a lot of questions from the audience, but due to the time limit, I think we stop here. And thanks Bing for the beautiful talk. Okay, thanks. Bing.
Thank you. Thank you very much, everyone.
Thanks. Okay. So our next talk is from Professor Wei Hong. Dr. Hong is currently an associate professor of neurobiology and biological chemistry at the University of California, Los Angeles. And I think he will talking about understanding the social brain. Please, Weizhe.
Thank you very much for the introduction. And thank you for the opportunity to share with you some of our recent work. Let me get my slide up. Can you see my slide?
Yeah, no problem.
Okay, perfect. So I will discuss a very different topic, social behavior in animals and how it's regulated in the brain. We live in a socially constructed world. We interact with others regularly, whether at work, at home, or right at this moment. Social interaction also allows us to acquire knowledge and technology over successive generations, making humans the most intelligent species on Earth. To navigate the world around us, we continuously make decisions and take inputs and feedback from the environment. This creates a dynamic loop between us and the external world. As part of this process, our brain has evolved to form predictive models based on the statistics of the world. As the majority of the physical phenomena can be explained or approximated by a second-order linear system or simpler models, our adaptive actions and the underlying neural processes are built upon this largely deterministic nature of the physical world. By contrast, maneuvering a social environment engages the brain in a fundamentally different way. Interacting agents are not isolated. The output of one brain will serve as the input of another, and this essentially forms a dynamic feedback loop between two individuals. This dynamic and reciprocal exchange of behavior makes social interaction substantially more complex and unpredictable. How does the brain regulate this process and make appropriate social behavioral decisions? What are the underlying neural and computational principles? These are the fundamental questions that we are interested in addressing in the lab. Most animal behaviors, from feeding to mating and fighting, are driven by one's own survival and reproductive instinct and serve the purpose of benefiting oneself. At the same time, we've also seen extraordinary examples of altruistic behaviors. The existence of these behaviors has long puzzled evolutionary biologists since Darwin, and the human nature of these behaviors have been debated by philosophers for centuries. What mechanisms motivate humans and other species to behave in ways that benefit others, even at a cost to themselves? These behaviors are referred to as pro-social behavior, which can be loosely divided into three categories that aim to support three different types of need. Comforting alleviates the negative emotional state experienced by another individual. Targeted helping is directed towards someone who has difficulty completing a goal-directed action. And sharing supports others' material needs. All these behaviors, pro-social behaviors, have been observed across a wide range of social species. The display of pro-social behavior involves a very complex multi-step process. A bystander first needs to be able to sense and perceive the emotional state of others, and this may lead to an increase of stress level of the observer through emotional sharing, a process commonly known as empathy. And this may result, and this process in turn could motivate the observer to take active pro-social actions, such as providing aid or consolation to others. And this may result in a reduction of the stress state of others, which is then transferred back to the observer, eventually reducing the negative state of both parties. This shared relief can also provide an intrinsic reward for the helper which reinforces this behavior. In humans, the motives of prosocial behaviors can be very complicated and may involve other elements, such as the feeling of being obligated to help in order to return the favor that one has received, honors or gratitudes, or moral judgments. There are so many open questions and unknowns for each of these steps, and here I'd like to focus on one of the most critical steps, the regulation of prosocial actions. And more specifically, when the animal perceives the emotional state of others, how do they decide to engage in prosocial behavior towards others? What brain regions and neural circuits are involved to mediate this behavioral response? Inspired by the work of Larry Young in Prairie Voles, we ask how mice behave towards their partners when their partners are stressed. To do this, we perform a simple behavioral assay. We take a pair of co-housed animals and subject one of the animals to a stressor, such as foot shock, and examine how the other animal behaviorally responds to the stressed animal. In a control experiment, we separate the animals for the same period but do not stress the animals. During this assay, we observed a robust increase of allogrooming towards the stressed animals. Here's a raster plot showing allogrooming in pink, which is substantially increased towards stressed animals, compared to control conditions where partner animals are not stressed. This suggests that mice are capable of displaying increased allogrooming towards their stressed partners. Allogrooming is one of the most common forms of affiliative social touch in animals, which usually involves gentle touch or licking of other animals' fur or skin. In humans, affiliative social contact often take the form of gentle social touch, such as hugging, petting, and caressing. These forms of social touch provide a pleasant and calming experience to others, and is an important part of our social life. Indeed, we found that mice that receive allogrooming showed a reduced level of anxiety-like behaviors in the open field tests compared to animals that did not receive allogrooming. This suggests that allogrooming interactions in mice also produce a stress-relieving effect. We are very excited about this observation that mice are capable of displaying comforting behavior towards others, and this opens up exciting new opportunities to study the neural mechanism underlying this behavior. To study the neural mechanism, we ask two fundamental questions. How does the observer sense the emotional state of others during this process? And how is this emotional sharing transformed into a behavioral decision to evoke allogrooming behavior? In rodents, olfactory and pheromonal cues play a central role in their social communication. And we found that if we swap urine from a stressed animal and transferred it to an unstressed animal, we're able to elicit allogrooming towards the naive animal that received the odor. And this suggests that the olfactory cues from the stressed animals are sufficient to elicit this behavior. The medial amygdala, or the MEA, functions downstream of the olfactory system, and is important hub in processing olfactory and pheromonal cues. And our previous work, and that of Niraj Shah and others, show that the medial amygdala regulates several types of social behaviors, such as aggression, mating, and parental behavior. So we hypothesized that the medial amygdala, the MEA, may also play a crucial role in regulating allogrooming. Using a miniaturized microendoscope made by Hemant Ghoshani and Daniel Haroni at UCLA, we're able to image neural activity from hundreds of single neurons in the brain when animals engage in naturalistic social interaction in a freely behaving manner. Using this technique, we recorded single neuron activities in the MEA when animals engage in pro-social interactions, and we recorded from the brain of the subject animal when they interact with either a stressed or unstressed partners. Among the MEA neurons that we recorded, we identified different populations of neurons that respond differentially to either stress or unstressed animals. Interestingly, MEA neurons respond more strongly to stressed animals, and this raises an intriguing possibility that the MEA may play an active role in mediating the behavioral response towards stressed animals. Indeed, we found that the fraction of MEA neurons are specifically active during allogrooming, but not other social behaviors such as sniffing. In addition to single neuron responses, we also examined population responses of different behaviors, and we found that the population activity during allogrooming is also distinct from those of other behaviors. This suggests that the MEA neurons not only respond to other animal stress state, but also encode allogrooming behavior. One of the main advantages of dissecting circuits in mice is the availability of genetic tools to label and manipulate genetically defined cell types. So to better understand the cellular organization of MEA neurons, we first performed single-cell RNA-seq. By sequencing about 45,000 single cells from the MEA, we identified a number of neuronal cell types that are marked by different neuromodulator genes. Here, we focus on three major neuronal cell types that are marked by the expression of three neuropeptide genes, TEC1, SST, and CCK. TEC1 is also known as substance P. Using in situ hybridization, we confirmed that there's limited overlap among the three populations. So in order to test the function of individual cell types, we take advantage of available Cree lines, and we are able to use them to selectively label each of the three subpopulations and activate them using optogenetics. And strikingly, we found that activation of TEC1 neurons, but not SST or CCK neurons, promote allogrooming. This is the optogenetically induced allogrooming, which is time-locked to the photostimulation period. To our knowledge, this is the first evidence that activating a select population of neurons in the brain can directly promote comforting behavior. We've also found that optogenetic silencing of TEC1 neurons can acutely suppress allogrooming, suggesting that TEC1 neurons are also required for this behavior. Interestingly, activation of TEC1 neurons also elicited self-grooming, a different form of grooming that's directed towards the animals themselves. So we hypothesized that this could potentially be due to the expression of TEC1 in a subset of glutamatergic neurons that have been previously shown to promote self-grooming behavior. So to test this possibility, we use an intersectional genetic approach to specifically target the GABAergic subpopulation of TEC1 neurons, and we ask if this population can selectively activate allogrooming. So to do that, we use a Cree and FLIP double-dependent channel rhodopsin and express it in a VGAT FLIP and a TEC1 Cree double transgenic animals. And this approach allows us to specifically label and activate a very select population of neurons that are defined by the cell type, the expression of cell type markers. And strikingly, activating this very select population of double positive neurons only promoted allogrooming, but not self-grooming. This suggests that TEC1 GABAergic, the GABAergic subset of TEC1 neurons represent a specific population of neurons that drive allogrooming behavior. We next examine the function of other intersectional populations, using a Cre-on flip-off strategy, we specifically activated the glutamatergic subpopulation of type 1 neurons. And as we predicted, it only promoted self-grooming, but not allo-grooming behavior. This means that the type 1 neurons can be separated into two different subpopulations of neurons within the GABAergic subpopulation, and that they're responsible for different type of grooming behaviors. The GABAergic subpopulation is responsible for allo-grooming, and the glutamatergic subpopulation of type 1 neurons is responsible for self-grooming. I mentioned earlier that the MEA is also involved in aggression. Using a Cre-off flip-on strategy, we specifically activated the type 1 negative GABAergic neurons. And using this strategy, we found that it preferentially evoked aggression, but not allo-grooming. This suggests that aggression and allo-grooming are controlled by distinct subpopulation neurons within the MEA. The medial amygdala project their axons to multiple different downstream areas, and the medial preoptic area in the hypothalamus, where the MPOA is a major target area of the type 1 positive GABAergic neurons in the MEA. So to test the functional role of the MEA to the MPOA circuit, we performed two sets of experiments. In the first experiment, we injected retrogradely labeled double-dependent channel rhodopsin into the MPOA and placed the optic fiber in the MEA. This allows us to manipulate MPOA projecting type 1 positive GABAergic subset of neurons. And this is a very select population of triple positive neurons within the MEA. And we found that activating the select subset of MPOA projecting neurons is sufficient to promote allo-grooming behavior. In the second experiment, we directly activated the axonal projection of these neurons in the MPOA, and it produced a similar phenotype. So together, these data indicate that MEA type 1, the type 1 GABAergic neurons in the MEA promotes allo-grooming through their projections to the MPOA. Together, we show that mice are capable of displaying comforting behavior in the form of allo-grooming. This opens up exciting new opportunities to dissect the underlying neural circuits and mechanisms using genetic tools in mice. We found that MEA neurons not only encode the stress state of the other individual, but also the allo-grooming behavior. Taking advantage of single-cell RNA-seq and cell-type specific manipulations, we identified a direct causal role of a specific subpopulation of neurons within the MEA, the type 1 positive GABAergic neurons that regulate allo-grooming behavior. And this effect is mediated by the projection of these neurons to the medial preoptic area in the hypothalamus. One working model is that this MEA to MPOA circuit may process social sensory inputs that represent partner animals' stress state and transform the signal into a behavioral decision to evoke allo-grooming. We're very excited about this finding because it provides an entry point for us to further dissect the role of other downstream and interconnected brain area in comforting behavior. Pro-social interactions represent a great example of how social cues and behaviors reciprocally influence the behavioral internal state of each animal. Indeed, as I mentioned in the beginning of the talk, the very definition and the unique feature of social interaction is that it happens between two individuals. We can think of the interaction between two animals as an interaction between two brains or two high-dimensional neural networks. So far, we as a field have largely focused on a single brain and how the single brain processes social information and reacts to it, as if the social inputs from the interaction partner is always static. But this approach may miss important aspects of social behavior and may not fully capture the dynamic nature of social interaction. So in order to study neural computation in the brain, we need to know the input and output space. But our natural communication and interaction are only observable at a very low dimensional space. For example, when we study mice where olfactory pheromonal information are their primary sensory cues, we are actually unable to assess the odor space in a spatially and temporally resolved manner. Another limitation is that there is no trial structure during naturalistic social interactions, and they present a challenge to compare neural signals across events or experiments. An alternative approach is to use a multi-brain perspective and treat two interacting brains as a single system. By accessing neuroactivity in both brains at the same time, we may bypass some of the limitations such as the neural information from two brains share the same timeline. And this not only allows us to better understand the neural computation within each brain, we can also ask whether neural behavioral and neural properties may emerge at the level of couple system. So from this perspective, we image neuroactivity in two animals simultaneously when they engage in naturalistic social interactions. And here we focus on the medial prefrontal cortex, or the MPFC, which is important for many behaviors, including social behavior. And here we examine neuroactivity in the medial and when we examine neuroactivity in the medial prefrontal cortex, we observe something really surprising. We found that the activity from two interacting animals become more correlated during the interaction, and the correlation is substantially above chance and above the situation when two animals are not interacting with each other. And this correlation is not due to two animals sharing the same physical environment or receiving the same sensory inputs. When the two animals are still in the same environment but are separated by a divider, the level of correlation is substantially lower compared to the animals interacting directly with each other. And this suggests that correlation across brains require direct ongoing social interactions. This phenomenon of cross-brain correlation has been previously observed in humans. This is the very first study where researchers use fMRI to image two people when they play computer games, and the signals in two brains become correlated. Michael Yartsev's lab at UC Berkeley also shows that bats also display correlated brain activity during their social interactions. So a critical question for the field is how interbrain synchrony emerge from single cells and neural circuits. This is a question that people have not been able to address in the study of humans and non-human primates. There are two possible explanations for how interbrain correlation arises from single cells. One possibility is that synchrony arises from uniform activity across all the cells. Alternatively, synchrony may occur between specific subpopulation of cells in different brains. To distinguish these possibilities, we take advantage of single neural imaging at single cell resolutions, and we found that MPFC has separate population of neurons that encode either one's own behavior or the behavior of the interaction partners, and we call them either self cells or other cells. And we found that if we computationally remove either self cells or other cells, we significantly reduce the interbrain correlation. These data indicate that interbrain correlation arises at least in part from cells that encode self and others' behaviors. One way to think about it is that if I have neurons that encode my own behavior and your behavior, and if you also have neurons that encode your behavior and my behavior, we basically have neurons in both brains that encode the common repertoire of all behaviors between us, and thus the overactivity of our brains become correlated. Another question is whether this approach can tell us more about social interactions. When we look at the moment when only the dominant animals behave, we found that the level of correlation across brains is substantially higher than the moment when the subordinate animals behave. And this suggests that when you have a social relationship where one animal is more dominant than another, the more dominant animal exerts a stronger influence on the intercorrelation between two brains. We are also able to show that the level of correlation between two brains predict how animals behave in the future. We found that if two animals are more synchronized with each other, one animal is more likely to behaviorally respond to another animal's behavior in the next moment. It also predicts the difference of their social dominant status. These data together suggest that interbrain synchrony may serve as an emergent neural property across animals that predict how animals behave in the future and how they develop their social dominant status. Together, our results suggest that interbrain neural synchrony, which arises from during social interaction, emerges from neurons that encode self and others' behaviors. And this also, we also found that interbrain correlation may serve as one of the emergent neural property of the couple system that can predict how the system behaves and evolves over time. Although this is just the beginning of our effort to study social interaction using a multi-brain framework, our findings suggest that we can learn more about social interaction by accessing neural activities in two interacting animals at the same time. With that, I would like to end here and thank the people who have done the work. The work would not have been possible without talented people in the lab, in particular, Emily, James, Lyle, and Shan, who have done the work presented today. I'd also like to thank our collaborators and all the generous funding sources. Thank you for your attention. I'm happy to take questions.
Okay, thanks, Weather, for the excellent talk. So, any questions?
Hello, Professor. I'm Wang Yan from Peking University. Thanks for talking. Very interesting studies. I have noticed that these studies are based on mice. We know that humans have more complicated situation because of more complicated behavior and cognition. Are there more experience showing that the results in mice are consistent with those in humans? Thank you.
Yeah, that's a great question. So yeah, and it's obvious that humans and mice have very different repertoires of behaviors and they interact very differently. And there's no question about it. But certainly mice also display pro-social interactions to help each other. And from a conceptual level, that's conserved. And the advantage of mice is that we have genetic tools that allow us to dissect circuits and understand how different circuit components are involved during the selection of different behavior, the processing of different social and sensory inputs. So I think from that perspective, it's important to understand what's going on in mice and how different circuit components contribute to behaviors in mice. And certainly we have to acknowledge and understand the potential differences between human behavior and mouse behavior. And hopefully one day we can find out what the mechanism is in human brain and how circuits in human regulates human behavior.
Thank you.
there is a question in the chat. What would happen if the synchronized network is disturbed? What will be the resulting behavior? So we don't know yet. So we have not performed perturbation experiment to disrupt synchrony. To some extent, synchrony reflect the ongoing social interactions. It may not have a causal role in driving social interactions, obviously there's no physical connection across brains, but synchrony as a phenomenon could potentially reflect underlying neural processes such as mutual attention or others, and the neuroactivity that are related to synchrony could potentially drive social interaction. Maybe not the synchrony itself, but the neuroactivity that are synchronized. So we are certainly very interested in disrupting the activity during synchronous activity, during the synchrony process, and ask whether that's required for ongoing social interaction.
I have a question. So when you're showing that the GABAergic type 1 positive neurons are responsible for the allogrooming, I wonder you need to activate most of them, or just a very small percentage is enough to make the allogrooming achieved?
Yeah, that's a great question. So we found that within the GABAergic neurons, we only need to activate a small subset, which is TAC1 positive. So we found that a very small subset of this TAC1 positive GABAergic neurons is sufficient to drive allogrooming. But even within that population, we believe that not every single neuron is required. We don't need to activate every single neurons within that population to drive allogrooming. Only a subset is sufficient. But what
about the number? So you in total need just activate, for example, 100 these type of neurons is enough, or you need to activate several thousand?
So in our experiment, we activate the neurons that are on the order of, I believe, several hundreds. We didn't try to activate fewer neurons, but yeah. So there might be a minimal number of neurons that are required to drive the behavior, because we don't know exactly what that number is. So we do know that if the virus is not very well expressed, then we don't get a behavioral output. So that do suggest that you need some number of neurons to drive the behavior. But it's sort of hard to determine exactly how many neurons are required.
Hi, Weijie. I have one question. I think this is for the behavior study with synchronization is a really unique opportunity to understand the human behavior, right? So from your study, do you have any clue or molecular clue or cellular clue or evolutionary clue for how this synchronization get involved during the long history of the animal evolution?
Yeah, certainly. The brains are well conserved. There are a lot of brain regions that are conserved that serve some of the important brain functions, including decision making, including social behavior. So at the level, we have brain functions that are quite conserved. So molecularly, it's hard to know. So I think when we were talking about genes and gene expression, epigenetics, and all the way to behavior function, there are a lot of intermediate steps, including the expression of those genes in specific cell types, the function of specific cell types during a particular behavioral context, how those cell types are organized together into a functional network, and how do those functional network sort of behave in a particular behavioral context. So we are talking about multiple layers, and it's sort of hard to draw links directly between the two. So certainly there are specific genes that are involved in defining individual cell types. Whether those genes are involved in functionally for those behaviors still sort of unclear and needs to be tested. But I think that's a great question, and certainly we have limited genes, and genes are very conserved. I wouldn't be surprised to see that some of the genes will mark specific neuron types that drive synchrony, and that we are very interested in finding that. There are a couple questions in the chat. Do you want me to answer those, or should I answer?
Yeah, please. Can you first read it and then give the answer? Thanks.
Sure. So there's a question about sex differences, whether there's sex difference in the allogrooming behavior. Is if the pair is strangers, do you see similar level of allogrooming behavior towards stressed ones? So yes, there is sex differences. For some reasons, male display somewhat higher level of allogrooming towards their partners, but I would say the comparison is a bit difficult because currently we're using same-sex animal pairs, so which means that if we test males, they would display behavior towards another male. If we test a female, they would display behavior towards another female. So you may also have differences in how males and females respond to a stress cues. They may reduce, they may release a different amount of stress cues that can be perceived differently. So yeah. So the short answer is that we don't know, but there are some apparent sex differences. So in terms of familiarity, yeah, there is a familiarity bias in allogrooming, as in most of the pro-social behaviors in rodents, other animals, and humans. So animals display higher level of pro-social behavior between familiar animals, and this is the case in allogrooming in rodents. So familiar animals, especially co-house partners, will display substantially higher level of allogrooming compared to strangers. And there's another question about activating the GABAergic TEC1 neurons in the MEA, and whether we see a change in stress level of mice, whether there's any effect on other social behaviors such as mating and aggression. So we did not see a change of stress level in the subject animals. And when we activate TEC1 positive GABAergic neurons, we didn't see other behaviors such as mating and aggression. But I did present earlier that if we activate the TEC1 negative GABAergic neurons, we preferentially activate aggression. That does suggest that aggression and allogrooming are controlled by potentially separate population neurons within the MEA. Although I should clarify that even though the TEC1 positive and TEC1 negative neurons have preferentially different roles in different type of social behaviors, but it doesn't mean that that all the TEC1 neurons are homogeneous. It's just one population, and every single one of them is involved in allogrooming behavior. It could still be a heterogeneous population. And the question that Fucho asked earlier is whether how many neurons are involved. We don't know. So it could still be a heterogeneous population, and it could potentially be subdivided. And even a small population of TEC1 neurons may be involved in aggression, but just that the predominant effect of activating the entire population is allogrooming. That's a great question.
Okay, so I think due to the time limit, let's thank Weijia again for his excellent talk. Thank you, Weijia.
Thank you.
Thank you. So let's move on. So our next speaker is Professor Xiaoqun Wang from Beijing Normal University, and also Xiaoqun also from the Institute of Biophysics, Chinese Academy of Sciences. I think today Xiaoqun will talking about his excellent work about exploring the development and evolution of human brain with single-cell multi-omics approach. Xiaoqun, please. Xiaoqun.
Yes, let me share my screen. I have sort of like a challenge to share my screen. What's going on here? Desktop. Can you see? Yeah, can you see my desktop?
Not yet. Maybe wait several seconds.
Just a second. Let me see.
Long Kai. Long Kai is a professor of biology and biological engineering at Caltech, and Long's lab works in the field of spatial genomics by imaging transcriptomics of cells within their native tissue spatial context. And Long is really a pioneer in the field to do the spatially resolved single cell transcriptomics analysis. And his strategy is really excellent and make a lot of beautiful biological discoveries. I think today Long will talking about spatial multi-omics, RNA and DNA seq-phase class. So Long, please.
Okay, well, thank you, Fuchao, for the introduction. It's really great to be here, and it's actually been a really a lot of fun in the last hour and a half or so. My kids are paying rapt attention to the talks, and it's been super easy with them actually tonight because they're paying so much attention to the talks. Anyway, so thank you for the invitation, and I want to talk about spatial genomics and both RNA and DNA and protein, and looking at all of these features in the single cells. And so, just for disclosure, I'm a co-founder of a company called Spatial Genomics also. As all of you here probably know already, that single cell genomics has really transformed biology in the last decade or so, starting with the pioneering work of Fuchao. And and many aspects of biology, it requires single cell resolution as well as spatial resolution to resolve the patterns in the tissues as well as the interactions between different cells. So this is really true in the brain as well as in embryos and many kinds of tissues. And so, as people know, the spatial technologies are really very rapidly becoming mature. And, generally I think of them as three big groups. One is in situ sequencing that's pioneered by Mads Nielsen and then George Church and various other people. The other one is slice-seq, where you literally put a piece of tissue on a patterned microarray and then lyse the tissue and then spread them out, capture the RNA. So this is spatial transcriptomics, slice-seq, and now more recently stereo-seq and so on. I'm going to focus mainly on the multiplex fluorescence in situ hybridization approach, FISH, that we developed 10 years ago. And this is a method that has the highest sensitivity and also preserves subcellular resolution. And so that gives you a lot of information about how the cells talk to each other that are missed in other methods. So this is fluorescence in situ hybridization is based, single molecule FISH is developed actually almost 20 years ago by Ralph Singer and then Arjun Raj. These are the pioneers in the field that showed that you can use these short oligonucleotide probes to hybridize against transcripts in single cells. And then we hybridize these probes and put them on a microscope. You can see these bright dots that correspond to the individual transcripts in the cells. And you can count the transcripts with almost absolute quantitation. This is considered the gold standard method in the field. It detects basically 100% of the transcripts. But then, if you want to multiplex this to a very large number of genes, to the transcriptome, as we were thinking about when we started this, that faces two major challenges. One is that if you do single molecule imaging, you're limited to a few number of fluorophores you can look at a given time. So on a typical microscope, you only have 3 or 4 colors you can use. And then the other problem is that each dot here, as you can see this image, is diffraction limited. So they occupy some physical space in the image. So that's typically, half a micron or so. So if you have a cell, that's, let's say 10 microns in diameter, then you really can only fit in a few thousand of these dots in a single cell. And that's actually what limited the field for a very long time. So, the way we kind of think about overcoming these problems is using sequential hybridization and what we call seq-FISH, to use multiple rounds of hybridizations. And then and you can target the same transcripts over and over again, such as, for example, this transcript was blue the first time and then painted red the second time, yellow the third time, green the last time. And this gives you a barcode that allows you to distinguish all the different transcripts in the cells, or any kind of nucleic acids, right, or any target molecules. So we first demonstrated this with messenger RNAs with this paper. And with this scheme, then with 4 different colors, you can scale up to hundreds to tens of thousands of genes in just a limited rounds of hybridizations. So this is the first proof of principle experiments in cells. These are actually single E cells. And you can see that over multiple rounds hives, you can spell a particular sequence of colors for each transcript. And then you can look at a particular region of the image and then quantify the number of times a particular barcode occurs. That tells you what the transcript is. And shortly after that, we showed that this works not just in cells, but also in tissues. And this is targeting about 100 genes in mouse brain slice. And you can see there are many transcripts in the cell bodies as well as actually in the neuropils. If you focus on this particular regions here, you can see that these transcripts switch colors over multiple rounds of hybridization. So this guy goes from purple to red to yellow. And then we can show the detection efficiency even after multiple rounds of hybridization, stripping, imaging, and repeat. You can get very high detection efficiency, about 80% here. So that means if you have 10 copies of this transcript in a cell, you were able to, on average, get 8 copies per cell, and relatively low false positive rates. And then, shortly after that, we showed that, this is not just limited to hundreds of genes, because when you look at messenger RNAs at the level is only hundreds of genes, it's really good for confirming single-cell RNA-seq results, but it doesn't, you're not finding anything new really beyond that. So we wanted to scale this method up even from the beginning to the whole transcriptome level. And we first showed that this is possible by targeting intronic regions. And so if you fish against the introns, you see that they are mostly present at the in the nucleus at the transcription active site, because introns are very rapidly degraded as they're coming off of the nascent active sites near the chromosome where the gene is. So here's a single mouse nuclei, and then each one of these dots is a single transcription active site, and the white thing is nucleolus. So you can see all these nuclear features in the cells in addition to the transcription active sites. So we observe actually many interesting features by looking at these nascent active sites. First of all, they seem to be on the surfaces of chromosomes and also near interchromosomal regions. They're excluded from the nucleolus as well as DAPI regions. So it seems like these active sites live on sort of a 2-dimensional surface, and I'll expand upon this when we get to the DNA fish part. But this is one of the first measurements that show us that we can scale these seq-fish measurements, in situ spatial measurements, to the transcriptome level. Okay, but now if you want to target messenger RNAs, you still have this issue of overlaps between different fluorescent dots. So how do we overcome this? So, we've actually initially thought to try to solve this problem by using super resolution microscopy. And it turns out, super resolution microscopy is really hard to do. And, it only works on very flat cells, and, it just wouldn't work in tissues. And because you're detecting single fluorescent blinking on off, it's actually very hard to do this in any tissue that have autofluorescence background. So this was just a proof of principle experiment. Did 30 genes, and then it realized that actually this is not going to be a viable path forward. Another way to increase more space, essentially in a tissue, is using an expansion microscopy that was developed by Ed Boyden's lab. And here you physically expand the tissue, such that even if your object is diffraction limited, now you have more space to fit that diffraction limited object in. And the disadvantage of using the approach is that imaging time is actually a major limiting factor in most spatial transcriptomics experiments. So if you expand the tissue 4x in one dimension, that's effectively 64 times bigger volume. And so now you experiment previously, which takes, let's say, 2 or 3 days, will take now 100 days, right? 60 times longer. And that's just not a viable path for imaging very large tissues. So we actually realized that the seq-fish method directly gives you a way to overcome this density issue. And the way to do that is that if you call only a subset of the transcripts at any given time, then you can use this Gaussian fitting trick that to shrink the size of the dots in the image. So the dots look nicely Gaussian shaped in the raw images, and you can fit a 2D Gaussian to that dot shape, and then get a super localized position of the transcript in this case. And this way, over multiple rounds of hybridizations, you can build up a super resolution image. So that's what I'm showing here with a composite image here. And once you've done the composite image in super resolution, you can repeat this with barcoding and then generate the barcodes the same way as we discussed before. And so in one of the proof of principle experiments, we showed that by targeting 10,000 genes in fibroblasts, if you zoom into this little region here, you can see, in super resolution, all the different transcripts that are resolved in this region of the cell. So in this particular cell here, we were able to decode about 50,000 transcripts in the single Z layer. So there's at least 3 to 5 Z layers in the cell when we targeted 10,000 genes. So overall, this cell probably had on the order of 250,000, 500,000 transcripts that you're detecting a very large fraction of that in a single cell. And that's what, in this sort of color dot pattern, that's what you're seeing here. And we can do this not just in cell culture, but we can do this in brain slices. So, in the cortex, we can zoom in successively. You can zoom into this, layer 5-6 region, zoom into this cell, and then further zoom in, you get all the transcripts are decoded in the cell. And then if you zoom in here, you get the actual transcripts. They're detected in this region. So each grid here is about 100 nanometers. So you really are detecting these things at super resolution. So what you can do is you can get the transcription profile of these 10,000 genes for each of the cells in the different regions of the brain. We image cortex and also, as well as SVZ. And in the cortex, you can see going from the different layers, you get the different layer specific excitatory neurons, as well as the inhibitory neurons, different types, VIP, SST, the standard types, as well as different types of glia cells like microglia, astrocytes, and oligodendrocytes, and so on. And in the SVZ, you get many different cell types, and they follow sort of this transition between the neuroblasts all the way to something that's more mature, to the stem cells to the more neuroblast mature states. And you can see that they're patterned in this interesting pattern inside the SVZ. So what this method allows us to do is be able to get subcellular localization for all 10,000 transcripts in different cell types in the brain. And what we found is that many transcripts are, uniformly distributed in the cell, but there are many transcripts that are also polarized. So some of these genes that are thought of as almost marker genes for astrocytes turn out to be fairly polarized within single astrocytes. But then you also have transcripts that are more nuclear localized. So you get this global look at all the transcripts and their subcellular localization just in a single experiment. And then, we can apply this to olfactory bulb using the same 10,000 gene probe set. And when we do that, we can see there are many different cell types, the mitral cells, the tufted cells, and also you can resolve different types of this dopaminergic DH positive cells that actually wasn't resolved in previous single cell RNA-seq measurements. And because we know these are distinct types because they actually occupy different regions in the olfactory bulb. So that gives us confidence that these are real distinct types of cells. Okay, so I think the most important measurements you can get from these experiments, high resolution spatial transcriptome-wide measurements, is to get the cell-to-cell signaling interactions. So the ligand receptors, for example, in this case, by comparing the microglia endothelial cells, looking at what's upregulated, downregulated when these two cell types are next to each other, we saw there's a bunch of genes that are upregulated in microglia, and in particular this TGF-beta1 ligand with the corresponding receptors in endothelial cells that's not expressed in just endothelial cells or not next to microglia. So that suggests the interactions between these cells actually induces a specific pairs of ligand receptors to have these cells signal to each other. So I think that overall, going forward, this is actually the most important aspect of the things we can get from these spatial data is that you can't get with any other methods. And if you have resolution that's lower than subcellular resolution, you're just not going to capture these interactions very accurately. So since this initial demonstration of the transcriptome-wide Sequential Plus experiment, we've been applying this to many different systems. One of them is in the gastrulin embryo, and for example, here is an embryo slice and E8.5 embryo, and we can see all these different type of cells in this embryo that leads to different tissues. And what we found that was interesting was that this particular type of cells, the gut tube cells, actually broke into multiple clusters that happens a whole day earlier than people know about. So people know that 9.5 days, there are these different dorsal versus ventral gut tube cells, but then actually 8.5, we already see the differences. And then if you look at the differential expression patterns of these cells, you see many of the signaling factors like sonic hedgehog, cordon, and so on are actually differentially expressed in the dorsal versus the ventral regions. And so this suggests that these high resolution measurements really allow us to get a very detailed information about embryogenesis. So since then, we've done this in microbial biofilms. We have done this in other embryonic samples, and then as well as in many human tissues. So I want to switch gears and tell you a little bit about, looking at the DNA structures and looking at epigenetic states in single cells using this method. So I talked a little bit about messenger RNAs and introns multiplexing them, but we can apply the same methods to looking at many DNA loci at the same time, as well as looking at chromatin marks in the same cells. So, when you look at these fluorescent microscopy image of cells, this is a DAPI staining, for example, you can see there are, very heterogeneous structures inside the nucleus. There are DAPI-rich regions. These are typically heterochromatic regions. And then there are also DAPI-poor regions. And then as well as more, homogeneous looking regions. So exactly how is the chromosome arranged in the cells, right? And so we can, by multiplexing thousands of loci with DNA fish, we can actually see, this corresponding cell where all the 20 chromosomes are in this. These are mouse embryonic stem cells, how they're arranged in physical location directly in the cell. And we can show that these measurements actually matches very well with high-C measurements. So these ligation-based, chromatin contact information that have been talked about earlier. And so on top is the DNA Sequential Plus data at the chromosome scale at 1 megabase of the 25 kb resolution. You can see they match very well with what's shown with the high-C measurements. And in the same cells, we can measure, not only these DNA loci, which are showing these color balls going through different chromosomes, but we can measure many epigenetic marks using antibody fluorescence in the same cells. So these are the repressive marks, MAC-H2A1, and so on, and then we'll transition into that's nucleolus, and then the active marks, histone acetylation, speckles, and so on. So from this, right, all done in the same nuclei, we can generate integrated picture of how the chromosomes are arranged, as well as epigenetic marks. So by doing that, what you can do now is you can figure out, let's say for H3K9 acetylation, which DNA loci are closest to those chromatin marks. And that's the profile you generate at the 1 megabase resolution, I should say. And surprisingly, across this chromosome 11, you get this map. It actually is very consistent with what you get with a ChIP-seq measurement. So if you look at individual cells, you can see that actually these peaks occur consistently across many different cells, over hundreds of cells. these contacts between the DNA, these particular loci, right, on the chromosome 11 is actually almost consistently associated with H3K9 translation. And when we look at the data sets for different chromatin marks, this is SF3 is a mark for nuclear speckles. It also matches what people have seen using, for example, Sprite with DNA loci associated with speckles. And again, it's highly consistent between different cells. So now, because we have the imaging data, we can go back and directly look at where these loci are with respect to the chromatin marks. And so, the red dots, the purple dots are the DNA loci, and then the blobs are the antibody marks, antibody immunofluorescence, that shows you where the nuclear bodies are. And what's really interesting, I hope you can see this, is that these DNA loci are consistently associated between different cells, across many cells, are actually almost always on the surface of these nuclear bodies. They're not in the interior, they're not scattered around, but there seems to be attached, coated to the 2D surface of these chromatin marks. And so this is true for active marks, this is true also for repressive marks. The green is H3K9 transmethylation, and the fixed loci associated with them are highly consistent. There's a different cell here. You can see that, again, the same features. The green dots are associated with the green blobs. The purple dots are associated with the purple blobs. And then the same for these other marks, H3K9 isolation, K27 trimethylation. This is a very consistent feature that seems to be that many of the DNA loci, it's not like a spaghetti ball. It's more like, they're living on like this two-dimensional surface. It's almost like inside of a baguette, right? There are a lot of holes where rather a lot of compacted protein regions, and the DNA is sort of coating on those protein regions rather than the DNA themselves being on this compact ball. So this is just another cell to convince you that, this is highly reproducible. We can see this across many cells. And so, when you typically think about this ChIP-seq results, right, we see these peaks in this one-dimensional traces, but perhaps the picture is that many of these peaks are actually, lying on this two-dimensional surface on nuclear bodies. So that's the picture. And what's interesting is that if you look across the chromosomes, there are these loci that are consistently localized with speckles and then as heterochromatins. So in the same cell, right, chromosome 4 will span speckle and heterochromatic regions. And so even though, if you look at the physical position of the speckles and the nuclear heterochromatin, they look kind of randomly distributed in single cells. And if you look at the structure of the chromosome by itself, it also looks kind of random between different cells. But if you look at them together, you see that it's highly consistent between different cells. So for example, in this cell, you will have the same regions associated with speckles, and then the end of the chromosomes are associated with the speckle. So that part is highly deterministic between different cells. And this is really the advantage of doing these integrated measurements with many different proteins and DNA at the same time that gives you this information. So just to show that we can extend this assay to looking at mouse brain slices, we can image these regions. We can also actually look at RNAs, DNA, and immunofluorescence all at the same time in the brain. We can identify all these different cell types and look at the chromosome structure. So I think that was really interesting is that, if you look at the major cell types, they are pretty well distinguished by their DAPI features already, without looking at anything else, without looking at the RNA, without looking at the immunofluorescence, just the different features of the DAPI, the intensity of the DAPI, these different metrics. You can actually cluster the major cell types. So I think this is why, when you talk to a neuroscientist, they can look at an image of a brain slice and they'll tell you which type of cells they are. Just the physical morphology of the cells is highly cell type identifying. But then, of course, you can do all these measurements with antibodies and DNA sequence. And we see actually the same kind of features in the brain as we saw in mouse ES cells. There are these fixed points in the brain that span many different nuclear bodies, and this feature seems to be conserved in many different cell types in the brain. Okay, so I just want to tell you a very last thing, which is that there's another tool that we developed called Memoir that uses CRISPR editing to edit a scratch pad that we engineer into the cells. And this allows us to continuously edit the scratch pad such that over the course of many generations, you can track lineage events in single cells. And then this barcode can eventually be expressed as a messenger RNA and then measured by seq-fish along with the endogenous transcripts. So that's what's shown here. You have a linear tree, and then you express them as transcripts and then probe them, and you get in situ readouts of both transcriptional states as well as lineage information. And so this is a demonstration of this by reading out multiple, these scratch pads. Here in cell culture, we can read out the different barcodes. And then from the barcodes edited patterns, you can reconstruct a lineage. And then from that, you can get this tree. So, showing this that we can do this not only in cell culture, but also in fly brains. We did this, by generating a transgenic fly, which we call deepened mammalia. And then, over multiple generations, we can track the lineage, as well as mark out different cell types in this fly brain here. And one of the really interesting things we observed was that, cells that have the same lineage usually tends to be clustered, but then actually there are also cells that have very dispersed lineages, and they contribute to many different cell types. So cells that are spatially close don't necessarily have to come from the same lineage, but sometimes they do. I think the overall picture is that we can get these kind of informations, the integrated multimodal informations about lineage, DNA, epigenetic patterns, as well as transcription profiles, all directly with these image-based spatial transcriptomics. And this will help us answer many questions about how the cells talk to each other and how cell-based decisions are made. And, for neuroscience, I think, you can easily imagine that you can integrate this with calcium imaging and other types of activity measurements, and then look at the cell types and look at how different behaviors are cell type specific or not. Okay, so let me end here and thank people that did the work. So the DNA work is all done by Udai, who was just an amazing graduate student. And then the RNA seq-fish plus was pioneered by Linus in the lab, and Linus is a superstar, and both these guys are superstars. And these experiments would not be possible without them. And thank you very much for your attention. Happy to answer questions.
Okay, thanks Long for the really beautiful work and talk. So any questions?
Hello, can you hear me?
Yeah, please. Yeah.
I have some questions. The first one is that I've seen that you said the fixed point is located on the surface of the nuclear bodies. I wonder if it is possible that those fixed points were more accessible than other areas, that the signals were enriched in those fixed points.
Yeah, that's a very good question. I mean, that's something we were quite concerned about from a technical perspective. We showed that actually there are regions that are inside nuclear bodies as well that we can detect, and those things usually tend to be repetitive regions. And so it's not that the inside nuclear bodies they're inaccessible. It's just that the non-coding, non-repetitive sequences tend to be on the outside of nuclear bodies.
Okay, thank you. The second question is that have you applied the integrative work of DNA, RNA, and the nuclear architecture on genomic in stable cells like cancer cells?
Yeah, we haven't done that yet, but it's definitely a very interesting area to look at.
Okay, the third question is that I've read the paper you published on Science about the integrative work of DNA, RNA, and the nuclear architecture, and I wonder that the result you said that cell type specific interchromosome interaction and the transcriptionally upregulated loci between cell types tend to show closer interchromosomal distance at the populational level, does it suggest that the nuclear architecture and the epigenetic modification at the proximal point may be the prerequisite for the cell identity?
Oh, that's a hard question to answer. Like, which one happens first, right? I think that's what you're trying to ask, right? Like, does the spatial organization happen first in the nucleus, or does the cell type identification happens first? Is that basically the crux of the question?
Yes, I wonder whether if it's their establishment that facilitates the co-expression of cell type specific markers.
The co-expression of cell type specific marker. Wait, are you saying the other cell type specific markers tend to be close to each other physically in space in the nucleus? Is that the question?
Yes, I wonder if it's the establishment of the nuclear architecture and the epigenetic modification first, and then the co-expression of the cell type specific markers were facilitated.
Yeah, I think that's a really good question that I don't have the answers to, because we're looking at adult tissues. So I think it's a better, you can try to answer that question by looking at developmental processes, right? Like, and then see whether which process happened first, whether the RNA is there or the spatial organization is there. And we haven't done those experiments, so we don't know the answer. But if you look at the adult, terminally differentiated cells, right, in the mouse brain, they've been there for months, right, in this animal. So, you just can't tell which one happened first, right? Because the SST neuron is not going to convert to something else later on. You're just never going to catch that. So I really cannot answer that question. But I think it's a very good one. Which one happens first, the expression or the chromosome structure? If I were to guess, I would guess the expression happens first.
Okay, thank you. Thank you very much. That's all.
Thank you, Professor. It's quite interesting. And I wonder, can the dynamics of a single molecule be recorded as a movie in response to different stimulus?
Yeah, so these are all done in samples, right? You have to do multiple rounds of hybridization. And if you have a living sample, first of all, the probes can't get in, the second, they will start moving, and then you are not going to be able to decode the transcripts. So this is not done in live samples, not done in live cells, but you can get time points, right? Just like the same way you can get pseudotime in single cell RNA-seq measurements, you can do sort of reconstruction of trajectories using this kind of data. Sorry to disappoint you that this is not live, but then, we're talking about generating, terabytes of data already. And with fixed one-time point measurements, if you had live information on top of that, you'd be generating, a very huge amount of data.
Thank you. And another question is that, can this technique be used to study the effect of measure control, morphology, and location on cell function in different cells of the same cell type?
You definitely can get morphological information in the same sample, right? That's the benefit of these spatial imaging-based measurements. That you get these features, you get many features at the same time, so you get the molecular information. So that's definitely would be something that's interesting to explore going forward.
Thank you. That's all.
Thank you, Professor. I want to ask a question about the sick-fish data. As in a slide, there in some region, the cells are so close, and some information of the mRNA just between two cells. And so, how do we define the information belong to which cell? Especially when we have to analyze so many data from so many cells. Thank you.
Yeah, so that's a technical question about self-segmentation in the images. There's been a huge amount of development in that area in the last couple of years. So there are now very good algorithms that you can use, for example, DAPI, and as well as cell boundary markers to segment the cells automatically. And then you can group the RNAs inside those cells. Of course, there's always ambiguities, and you have segmentation errors, just the same way you have doublets and so on in single cell dissociative measurements, right? So, there's always a challenge, but, I would say 80-90% times you get the right cell boundaries, then you get most of the data that's right. So yeah, it is a complicating factor. And in some tissues, it's more challenging than others.
Thank you, Professor.
So I have some questions. So when because now your seq-phase plus the technology is powerful, but it's still only the expert can do it. So I wonder when your seq-phase will be commercialized, then every lab can use it.
Oh yeah, we can have that conversation offline. We're happy to help you guys get it working.
Okay. So another question is that when you do this spatially resolved transcriptome, the raw data is quite often is huge, right? You say it's terabytes data generated. So I wonder, just is there any way to organize to how to deposit these raw data into the public domain, because it's very useful. So if you only process data there and then throw away the raw data, it's really a pity.
Yeah, I mean, we hope to get it to a point where it's like the Illumina sequencing data, right? Where you throw away the raw data and then you just clear the process. But it's still, we're saving a lot of raw data, and it is a major challenge. I mean, I don't know. I don't have a good answer on how, we can try to compress the data in some ways that preserve some of the information, but not at the full bit depth that we take the raw images at. Like, if the images are full 16 bits, then maybe it doesn't need to be that.
And another question is for your spatially resolved genome. So can you just one time to analyze all the 20,000 genes loci together, or you still can just see some of them, not all of them?
So in the experiments we published, we're looking at about 3,000 loci per cell.
Mm-hmm.
So it's not all the loci, but gives us a representative information.
So it's still just about 3,000 loci, not 20,000.
Yeah, at the DNA level, we're not doing the whole genome just yet, but yeah.
Okay. So I think due to the time limit, let's thank Long for the beautiful talk. Thank you, Long.
Yeah, thank you.
Okay. So now let's go to Xiaoqun's talk.
Okay, sorry. Yes, first I will apologize for the my computer difficulty, and I think it's because I'm also the co-host for this section, and my computer might just shut me down automatically to save more time for the expert talent talk. Yeah, I think it's a good choice. Yeah. Well, today I'm gonna show some of my lab's past work, and also getting support from Professor Fuzhou Tang. We sort of like collaborate for quite a while to integrate or in exploring the development and evolution of a human brain with the single cell multi-omics technique. Because it's everybody knows that during the evolution, there are huge the changes or variation compared the human brain to other mammals. But with limited technology accessibility, so there are very barely study on human brain, especially for human brain development. Since the introduction of the single cell multi-omics technique, so which allow us to get a little bit taste about how the human brain development, and also to get the information to integrate the evolution of the human brain while they get into larger and with high cognitive function. Obviously, everybody know one of the most remarkable feature of the evolution of the neural cortex is the enormous increase in the neuron number that reached its peak in the human brain. An enormous expansion in cortical surface area underlies the transformation from a smooth cortex to the highly to the highly folded pyramid neural cortex. This transition actually underscores the importance of understanding the process of neurogenesis in the developing neural cortex. So the human cerebral cortex have some unique genetic, molecular, cellular, and anatomical features which need to be further explored. Those studies have indicated the neurogenesis in the human occur in the great number of germinal zone and involve a large number of cell types and behaviors than in mice. So most notably, there's an expanded proliferating region termed the outer SVZ, what we call OSVZ, or some lab call BVZ, basal progenitor region in the developing human neural cortex that contains the majority of the progenitor cell during peak period of neurogenesis. First, we start from the prefrontal cortex developing in the human brain, since this is the most part of the important brain region in the human brain. So the many prefrontal cortex comprise a set of highly specialized brain region containing the beneath cell and serve as the center of the highly ordered cognition functions, such as memory, cognitive ability, decision-making, and social behavior. Dysfunction of the prefrontal cortex contribute to the cognitive defects and the majority of the neurodevelopmental disorders. Thus, there is therefore an urgent need for detailed knowledge of the development of the prefrontal cortex. With a single-cell RNA-seq technique, we could be able to identify cell type in the developing human prefrontal cortex and to explore their developmental feature. So actually, we analyzed at that time. We only analyzed about 2,300 single cells in the developing human prefrontal cortex from gestational week 8 to 26 using RNA sequencing. Actually, we can identify 35 subtype cells of 6 main cluster and tracing the developmental trajectory of this cell. We then reconstructed the developmental timing course and the lineage relationship using monocle analysis. And microglia and the interneuron were excluded because microglia are a mesoderm-derived cell, and the interneuron are generated in the ganglion eminence and migrate tangentially to the prefrontal cortex. So the remaining cell were distributed along pseudo-temporal order paths from neuroprogenitor cell NPC to neurons to O2 OPC and astrocyte, which is the glial lineage. To reveal the diversity of the neuro NPC in the human prefrontal cortex, we using monocle analysis. We found that the developmental trajectory of NPCs follow 3 major paths: the early and the later paths from ventricular radioglia, VRG, to the IPC, we call intermediate progenitor cell, and the later path to ORG cell, which we call OSVZ progenitor cell. We also found the twin peak of a neurogenesis pattern, which reached the peak at GW10 and GW16 respectively. So this result suggests the accumulated IPC at GW10 might originate from the ventral radioglia cell, whereas the majority of the IPCs, especially those located in the OSVZ, might originate from the ORG cells that are present at GW16. To validate this result, we immunostaining cortical of the prefrontal from GW8 to GW19. We observed early increases in the number of IPCs at GW8, just above the thick ventricular zone, and GW10. The number of the IPCs increased rapidly in the expanded SVZ. Thereafter, IPCs were not generated actively until GW16, when they were abounded in the ISVZ and OSVZ here. Through single-cell transcriptome profile of the sorted human neuroprogenitor subpopulation, we actually also identified primate specific gene, which we call TMEM14B, as a marker of the OSVZ. expansion of the TMEM40B in embryonic neural potential actually can induce cortical thickening and gyrification in the postnatal mice. To to further examine the TMEM40B function in cerebral cortex development, we generate a conditional knock-in mice in which human TMEM40B expression is driven by nesting creep. Well, actually, we found a conditional expression of TMEM40B in AMP consistently leading to the development of the enlarged brain and with the featured suture and the gyre in the newborn mice. We also can examine the structure of the suture and the gyre by staining for SETB2, meaning expressed in layer 2 and 3. We observed that the gyrific cortical plate in TMEM40B in conditional knock-in animal were enlarged when compared with the smooth control of the neural cortex of that exhibit the proper cortical laminin. To further explore the effect of the TMEM40B on deeper layer formation, we use a CTIP2 layer 5 marker and we observed an increase in CTIP2 positive deep layer neuron follow TMEM40B overexpression. So thus, we actually conclude that TMEM40B expression in the mice central nervous system could lead to the expansion of the SVZ and the appearance of the increase in ORG cell and RPC cell, thus ultimately increased both upper and deeper layer neuron to form the folded cortical with the proper lamination. So we also observed that the Paxis positive area expanded radially toward the intermediate parietal zone, which could be considered as the OSVZ-like region in the developing TMEM40B mouse neural cortex. And we also found ROS GGP activin like protein RQJP1 as a candidate associate protein. Overexpress of RQJP1 could phenocopy the effect of the TMEM40B overexpressed in mice. Suggests RQJP1 expression could induce an increase in the number of SOX2 positive potential, which display already like a morphology with cortical extension. Thus, we conclude that TMEM40B could drive the neural proliferation by increased phosphorylation and the nuclear translocation of RQJP1, which in turn promote G1 as cell cycle transition. So this data show that a single primate, a specific gene, could drive neural developmental changes that constitute or contribute to the brain evolution and development. So our single cell resolution data illustrated the complex diversity of cell types in the developing human prefrontal cortex that underline this sophisticated cognition function of human. So we found that accelerated neuron could be further divided into 3 groups according to their developmental stage. The group 1 is the GW8 to 12, the cell proliferation stage, and the GW13 to 16, the neuronal differentiation and the migration, and the GW19 to 26, which the neuronal function start to develop. We examined the electrophysiological property of neurons in different layers of the prefrontal cortex at GW26. We examined neuron from the cortical plate and the subplate of 3 GW23 prefrontal biological samples. In the upper neuron, which means the SETB2 positive and FOXP2 positive, action potentials were only barely initiated, with a low level of outward potential and the inward sodium channel currents. For the deep layer neuron, which is a SETB2 positive and FOX2B positive, it also exhibited stronger action potential and the pattern firing. There were no differences between whole cell membrane sodium and the potential current in upper or deep layer neuron, but the deep layer neuron exhibited more complex 3D morphology. In addition, both spontaneous EPSC, the excitatory post-synapse current, and the RPSC, the inhibitory post-synapse current, were detected in several deep layer neurons, suggesting that this cell were beginning to receive excitatory and inhibitory pre-synapse input. The subplate neuron also show more mature electrophysiology than the cortical plate neuron. Consistent with their mature electrophysiology property, the subplate neuron exhibit more complex 3D dendrite morphology than neuron located in the upper or deep limb. So gene enrichment analysis also show that node signaling were more involved in regulating the biological activity of neural potential cell than those of the neuron, and excited neuron development peaked at the GW16, and also whereas the interneural development peaked at the GW26. We therefore select neuron at the GW16 and the GW26 for pathway analyzed by gene set enrichment analyzed. So exon guidance signaling, including exon attraction, repulsion, and outgrowth, were more active in excited neuron at the early stage and more active in interneuron at a later stage. Suggests the cortical development is highly precisely programmed. So to further explore the neuronal development in the different regions of development of human cortex, we use a single cell ion sequencing to survey cells from different regions with the PON as a control. So the cerebral cortex can be divided into following lobes: the frontal lobe, parietal lobe, occipital lobe, and the temporal lobe, with each showing specialized function in sensory and motor control, and have a specific projection to different targets of the nervous system. In generally, the full brain region shares a similar developmental pattern as the pseudo-age evaluate of NPC in the four lobes of the cerebral cortex reveal a relatively late maturation between rostral and the caudal region. So for the occipital lobe is slower. So regional different regulation gene in each neurosubtype. We found NR2 F2 is highly in red, and it is in the TL specific gene in all cell subtypes. So neuron in the frontal lobe exhibit more complex morphology, while their counterpart in the parietal lobe show barely process. For the action potential with a mature kinetic could be invoked by a current stimulate in FL neuron. Although the resting member potential of the neuron in the FL was roughly equivalent to that of in the PL, and invert sodium current with a significantly larger amplitude was observed in the FL neuron. So this evidence demonstrates that neuron in the FL reach a more physiological mature state than do this in the PL at GW24. So as the also transit structure in the early brain development, the ganglion amylase gene generated dozens of different types of interneurons that go on to migrate throughout and weave together the developmental brain. Despite the projected development of the human cortical interneuron, we found that their diversity is specified within the ganglion amylase long before this cell reached the developmental cortex. So we also identified two populations of human interneuron with features that do not seem to be shared with the rodent. Prospective subtype of the MGU-derived fast-spiking neuron, and a large population of CGE-derived GABAergic interneuron in this here. So I would like to summarize here. We can decode the diversity and the molecular feature of the neural potential and the cell lineage in the development of human cortex with the single-cell multi-omics technique. And we can detail and analyze of the neuronal potential cells that with the new marker gene and a unique developmental of the intermediate potential cell. Expression of the TMM40B, the primary specific gene, could help increase the specific neural potential subtype and introduce the cortical gyrification. And we also illustrating the similarity and the heterogeneity for the different brain region within the different brain lobes. We also can identify the molecular feature of the human ganglion amylase and the interneuron, which is to suggest which can save the foundation for people to understand the diversity of the cell type contributed to the human brain development. So the second part, So since because of this accessibility and the remarkable ability to recapture the human embryonic neuronal development with the brain organoid, have this technique have step-by-step proved to be a valuable alternative way to current animal model for the study of human developmental. And they also have been used to as a model to study outer radionuclear progenitor population greatly expanded in the gyrification, gyrocephalic primate, as well as neuronal migration and circuit formation across the brain region. So we also try to set up the laminated cortical organoid in the past maybe 10 years. In the early version, we actually can see the typical human RPSC derived organoid grew faster and formed a ventricle-like cavity surrounded by neuron progenitor cell. Around this ventricle, a thick layer of SOX2 positive progenitor cell and some expanded 2G1 positive newborn neuron were observed in the organelle, indicating neuron progenitor cell and the newborn neuron generated in this organoid. This is a major but also there's major shortcoming of version 1 brain organoid technology is the lack of a ventricular system. So several apoptosis and necrosis occur within the organelle due to limited oxygen and nutrition delivery while surface diffusion. So this problem has hurdled the scientific to conduct various trials. To improve the oxygen and the nutrition supply and sustain the culture in the long term, it's a massive effort to introduce the vascularization to organoid. Thus, here we developed a 3D culture protocol to generate a we organoid by co-culture human embryonic stem cell or human RPS cell with a human umbilic when endosis pelvic cell in vitro. So by virtually by virtue of the prominent tube formation ability of a pelvic cell, we established an easy to follow protocol for reproduce generating vascular organoid by culture embryonic stem cell with the pelvics. Follow their schedule. We are complicated mesh-like tube-like vascular system can be readily introduced in several organoid. The long-term stability of the vascular system in V organoid was verified by its presence in V organoid culture for more than 200 days. We can see very complicated the vascular network within the organoid, and the tube-like structure is formed in the culture system. We observe that the HUVEC connect to form a mesh-like tube-like structure in V organoid. Here it is just 42 days. So vascularization in the V organoid primarily occurred just above the VZ and SVZ region, which is enriched with the SPY box transfer factor 2 radicular cell, and also HOPEX ORG cell and TBR2 RPC cell. So the vascular system mainly located in the outer layer of V organoid, which were enriched with the neurons, were sustained for over 20 days. So yeah, we also performed a Cas3 immunostaining in non-vascular organoid and vascular organoid, and found that the cell ratio of the cleavage Cas3 positive cell in V organoid was significantly reduced compared to that in the non-vascular organoid. In addition, we also performed immunostaining for hypoxia inducible factor 1, which is HIP1 alpha, which is a hypoxia marker. Consistent with the result of cleavage Cas3 staining, the cell ratio of the HIP1 alpha positive cell in the V organoid was significantly reduced. This suggests the vascular system really can help to the oxygen penetrated into the inside of the culture. So human brain microvascular endothelial cell, which we call HBMEC cell, as HBMEC cell play important role in the development of blood-brain barrier. So we examined that whether co-culture could induce the HUVEC cell to a more brain-like endothelial cell fate which can be reviewed by phospho GP, the glycoprotein. So given the differences between HUVEC and HB and EC cell, we also examined whether co-culture can introduce to adopt a more brain-like endothelial cell fate. The phosphoglycoprotein positive cell actually indicate that the HUVEC cell can step-by-step adapt to this brain-like endothelial cell cell fate. In the 42 days, the immunostaining also result for the dorsal progressive marker, ventricular marker, and the pan-protein marker in both regions in the 42 days actually show that clearly you can recapitulate the brain development in a different way, like protein cell and also the differential neuron. So further, we actually check whether or not this vascular organoid compared to the non-vascular organoid or compared to the human fetal brain tissue, whether or not they can recapitulate their cell type, cell fate, and also their developmental procedure. Actually, the high transcriptome correlation between the corresponding cell type in the organoid suggests there are no remarkable cell type difference between non-vascular organoid versus vascular organoid. But based on the transcriptome analysis, the cell type in organoid are also highly similar to the corresponding cell type in the human fetal telecephalon. The GO term enriched the protein cell in non-vascular organoid were response to hypoxia and positive regulation of cell death. This GO term are consistent with the highly number of HIF-1 alpha positive and cleavage caspase-3 positive cell observed in non-vascular organoid. Meanwhile, the GO term enriched in the protein cell in V organoid were positive regulation of the neurogenesis and the blood vessel morphogenesis. We also calculate the maturation score of the each cell in vascular organoid were control, with more vascular organoid cells than non-vascular organoid cell with a highly score suggests that with vascular system, the neuron mature faster compared to a non-vascular vascular organoid system. Also, the dramatic increase in outward current amplitude in day 18, but not the amplitude of the inward current, which suggests the neuron in V organoid might be more mature than those in non-vascular organoid, may accelerate the progress of the functional development of the individual neuron in vitro, which suggests the vascular system might not change the fate, but contribute to the neural maturation. In addition to the formation of the chemical synapse, we also investigate the formation of the collateral synapse, which means the gap junction, and in the vascular organelle, which we can use the dye or with the dual patch, suggests that they have a perfect chemical coupling and also the electro coupling within the system. The most striking thing is when we transplant the at 60 days, we transplant the vascular organelle into the host mice. Actually, you can see after couple of 1 month, 2 months later, actually you can see their gyro-like structure in this human embryonic stem based vascular organoid. They can develop some like a B2 positive cell. It looks like a gyrification formation in the host mice. And also we can see with the host, you can see a lot of like laminin penetrate inside the transplant organoid, suggests the blood vessel penetrate, the host blood vessel penetrate into this organoid system. And also most striking, we use the dye with the in vivo 2-photon imaging. We can monitor the blood flow actually flow into this vascular system in the organoid, suggests the host the blood system, vascular system connect to this transplant organoid. So we also can use this organoid to do the microcephaly disease modeling. Here we can see we use a human patient which have a ASPM mutation patient iPSC cell to generate the brain organoid, which you can clearly see their development smaller and also have a lot of defects, which can perfectly mimic the disease pathologies. And also we can use this vascular organoid to modeling the FCD associated epilepsy. You can see with the cultured patient vascular organoid, you can see the electro pattern is a perfect match to the human patient electro pattern when the epilepsy happened. So that's I would like to summarize here. So we can do with the constructing vessel system with the human cerebral cortex, and the organoid recapitulate the process of cortical development in vitro, and modeling also can modeling functional maturation with neurogenesis. We transplant with vascular organoid. We can recapitulate the vascular system in the mouse cortex, and also with less cell death of the graft. Also, we can use the organoid as useful to study the human brain development and also related disease. And I would like to stop here to really thank my lab member and also my wonderful collaborator in China. And also we have a sort of data, single cell data can be accessed in the onelaboratory.org. Welcome everybody to visit us. Thank you.
Okay, thanks, Jim, for the excellent talk. So, any questions?
Thank you, Professor. Can you hear me?
Yeah, please.
Yes.
I have several questions. And the first question is that previous research suggested that OL developed later than the FL, about 2 weeks. So the difference is doesn't be defined in the single cell data.
Yes, actually, we can, with the maturation score, we can actually clearly, our data show that according to the transcriptome profiling with the maturation score, we can see this is brain developmental differences within the 2 weeks. Actually, which is a little bit controversially, a little bit with another publication in Science, which is actually, when we merge these two data together, we can find our data is maybe make more sense. So yeah. So we verify that, and also with hopefully we can continue to verify that with staining with a new marker to do that. Yeah.
Thank you, Professor. The second question is that in your data, CG region just one kind of progenitor of interneuron. However, also in the 2021 in Nature paper from the Thomas J. Noaskiva, they claim that a kind of progenitor in the CG region also appear during the gastrulation week 15 to 18, can both generate exciter neuron and inhibit neuron. How do you think the relationship between the two kinds of progenitor?
Yeah, actually, following the Tom's paper, actually they do the not only with the single cell, but also with the transplantation, right? So actually, which is to suggest the which is kind of like a heavily debate whether or not the interneuron origin not only from the ventral part of the brain, but also they might have some clue actually originated from the exciter part of the brain. So well, actually, well, from one of our early data, we actually found a lot of NKX2.1, which is the interneuron plasma cell located in the early stage of prefrontal, which I think this is sort of like consistent with our data. So yes, while still we need more carefully to analyze whether or not in human brain interneuron where the interneuron origin, not only whether or not they are not only from the ventral part, might also from other part of the brain. So which is still I think still a puzzle in the field. We need to looking for more data to find the answer. Yeah.
Thank you, Professor.
So any other questions? So I have a question, Xiaojun. So now for the organoid culture, so do you because someone worried that the how to say the batch effect. So what's your feeling about the batch effect? It's very easy to control to similar level and make the different batches work very comparable, or you need to the batch is quite a severe issue, need to be very carefully controlled.
Yeah, this is a very good question. Actually, there are a lot of concerns from the early beginning about the culture system, right? Culture system. Actually, for the brain organelle culture right now, actually they do have two major methods. For one method, actually originated from Austria, like and also Lancaster from UK. Actually, they are non-supervised culture. So the brain organelle can develop all kinds of the diversity of variation. But right now, more and more people actually prefer the supervised culture system. So they actually can put some like a transfer factor or put some like a growth factor to supervise to the brain organelle differentiation into specific brain region or specific cell fate. For the supervised brain organelle, actually, the batch effect is much less compared to the non-supervised culture system. So it's and also since the human brain is so diverse, and also the brain region right now looks like they are also diverse and the variation among different brain regions. So people right now actually prefer to do the supervised culture and then pinpoint the different part of brain together for the fusion system to study the brain-brain interaction or brain-brain different brain region communication. Yeah. So for that part, the batch effects might not a big issue. Yeah.
Zixi, please.
Hello, Professor. Professor Wang, can you hear me?
Yes, please.
Okay. I have one question. As for the information of the astrocyte are so limited, and the astrocyte developed late. So I want to ask, compared to other cell type in humans, so how to assess the similar between the astrocyte from the organoid and from the brain organ?
Oh yeah, that's I think the question have a two part, right? Whether or not how to get access or analyze the astrocyte in the developing either human fetal brain or either brain organoid, right? For astrocyte culture in the vitro system, actually there need a very specific protocol to introduce the astrocyte to get the right ratio or get the right perfect ratio or the perfect number or perfect subtype. During the human brain development, actually yes, the astrocyte come up a little bit later compared to the excited neuron, but they share the my similar ancestor. So for that part of the research, actually right now more and more I think the lab actually pay attention to the study of astrocyte origin and also astrocyte diversity. Still, I think this is an open question for the field. For the in vitro system, right now we couldn't claim there are a couple of subtypes of the astrocyte we can develop or introduced in the culture dish, but whether or not it is fully recapitulate the in vivo development, still actually it's an open question. I think so, yeah. We encourage more and more labs to join in to study this question. Yeah.
Okay, thank you.
Hello, Professor Wang. I have a question that do you plan to perform some alternate control about drug screening for some specific disease such as Alzheimer disease, and what is the challenge and difficulty?
Yeah.
Thank you.
Yeah. For the Alzheimer disease, it's take a time quite long to develop the phenotype, and also to fully mimic the pathologist actually in vitro still is a challenge in the field. People use like a lot of methods, like even with actually patient-derived iPSC cell, actually might take almost a year to have some slight phenotype. So it's a challenge for the Alzheimer disease. But for the epilepsy, for the schizophrenia, for this kind of like disease, actually is easy to recapitulate the phenotype. Like for example, for the focal CD, the epilepsy disease, we actually can quite easily within the 40 days we can recapitulate the phenotype. So for that we could be easy to screen. Yeah, we don't have a large compound library, but we are right now actually trying to use some like a FDA approved drug to test whether or not we can find the new use for the old drug for this kind of screening. This is also a convenient way to do that. But for the still people need time or new strategy to help to force aged culture system. For example, actually there are like Lawrence Stoder in Memorial Kettering, their lab actually will try to set up some like a force aged system to study the brain disease like Alzheimer. Yeah.
Thank you.
Yeah.
Any other questions? Okay, I think due to the time limit, let's thank Xiaoqun for the excellent talk. So finally, I want to represent Professor Xiaoqun Wang and Professor Wensheng Wei to thank all the speakers for this panel, Professor Bing Ren, Professor Wei Zhehong, Professor Long Kai, and Professor Xiaoqun Wang for their excellent talk. They bring a lot of exciting new findings and also a lot of new exciting ideas to the field. I think now there's many different exciting cutting-edge technologies are applied to the neurobiology or the brain biology. So why people want to do that? I think because the neural system is probably the most complex system in our human body, and the brain is the most important and most complex organ in our body. So when you're using this new cutting-edge technology into it, and there will be unlimited chances and unlimited possibilities. So I think now is really an exciting era. People can using the best technology to answer these really complex systems, most exciting questions. So I think now is really a chance to join the field using your ideas, using the new technologies to answer the most challenging and most exciting questions. So I think as if you think deep enough and work hard enough, and very likely you will get exciting answers to the exciting questions in the field. And I think clinically, we can now address a lot of neural system related diseases. For example, for us to understand how it happened, and then how to diagnose it, and then how to cure it. And I think finally, the most ideal way is how to prevent the disease before you get ill. I think now it's a really exciting time. So let's thank the whole panel's speakers again, and hope you enjoy all these talks and then get some new ideas and new feelings about the field. So I think that's all. Thank you for your attention.